All right, ladies and gentlemen, welcome back. This is part one of the cell. Last day we took a little sneak peek into the different types of organelles. You guys looked at a few. We're going to go over them all right now. Just make sure that we know this is the goal for today. We're going to do a lot of work with the organelles and how they relate to each other in class, but out of this video, if you just have a general idea of what it does, I'm happy. So here's just a couple. All right, so let's look at it. First, we're just going to talk about the different types of cells. The first one is prokaryotic. What this means is these ones are usually a little bit more simplified, a lot of bacteria type, some protists. The main biggest thing here, no nucleus. It just has its DNA all kind of clumped together, but there's no membrane that surrounds it. With that being said, organelles that have membranes. So all of these other membrane-bound organelles that you're going to see next, these don't have. All these ones have is just pretty much ribosomes, which we'll see what the function of those are. And then it has its nucleus, the cell membrane, and the cell. So what is a ribosome? This is meant for protein synthesis, and we're going to see this again later. We've mentioned this already when we were doing our DNA unit. This is where the site, I guess we should say site of protein synthesis. It allows for the mRNA and the tRNA to bind and come together to position those amino acids. Plasma membrane. What happens here, this is in all cells, and this is meant to be semi-permeable. Permeable means, holes allowing things to come in. This is what controls what enters and exits the cell. The next one, difference or a certain thing about prokaryotic, these chromosomes are circular. As you're going to see, a lot of our chromosomes are kind of those X-shaped, and these sometimes are called plasmids, which we're going to see in the bacteria unit. And then finally, it usually has flagella, which is used for locomotion, which just means motion. Okay, so pause that if you need to write those down. Let's move on to the eukaryotic cell. And you can think about eukaryotic as being complexed, just like you are. Get it? You. So here's a eukaryotic cell. These are the ones that you guys were looking at on iCell with the iPads, all the different organelles here. So let's just look at a few characteristics of eukaryotic before we go into the actual cells. So these ones actually do have membranes. So they have these outer membranes that cover those organelles. It has a nucleus, check. These chromosomes are linear, like that. And how these ones divide is by mitosis. And this is something that we'll talk about a little bit later. Okay? So you can copy that down and moving on. So let's get into it. Let's talk about what each of these organelles are going to do. We know a little bit about the nucleus already because this is where the start of our DNA replication or transcription happen. So this is where chromatin occurs, and this is just kind of, this is DNA. Chromosomes is the organized DNA. It organizes into, DNA, into chromosomes when it's about to divide. Okay? And then in the nucleolus, which is in this is your cell, here's your nucleus, and you just have this. And it's a nucleolus, and this is where RNA and ribosomes are made. We have a double membrane envelope, so there's two membranes that are surrounding our nucleus. And the nice thing about them, they have pores, so that's where that DNA couldn't get out. So DNA, no leaving. And so we use the RNA in order to get out. The site of replication and transcription, which we talked about in our last unit, so that should be nice and easy to remember. And I'm going to give an analogy in each situation. So I have a blueprint here of a house. Why is this important for us to remember DNA? Well, because DNA is the blueprint, or it has the blueprint for all of our proteins. It has the DNA code, which changes to RNA, which will change to the order of the amino acids, just like this has the plans and the dimensions of the house that's going to eventually be built. Okay, so ribosomes, what are those? Same thing as we talked about before. This is all used in protein manufacturing, so protein synthesis, making a protein. There's two types. There's free and there's bound. If you have free ribosomes, it just means it's floating in the cytoplasm, which means just like the interspace between any other organelles. And this makes proteins, just like all the ribosomes do, that functions in the cell. Whereas bound ones, which means it's going to be bound to the ER, so the rough ER, which we're going to see in a second, this is for proteins used in the membranes, in cell membranes, and then also exported out of the cell for other reasons. Here is just a microscope picture of them. All of these little guys here are the ribosomes attached to the ER, which is here. Okay, and this is what the ribosome looks like, two subunits that come together. And I use the analogy that ribosomes are like chicken, because what comes out of the chicken is eggs. Eggs have lots of protein. Same thing, what comes out of ribosomes, proteins. Stupid way to remember it, but usually the ways that are the most unique will be the best ways that you remember it. Okay, endoplasmic reticulum, we call them the endomembrane system. What it is, it's, if you look at your nucleus here, it's a continuous with that membrane. So it's connected right to there. So you have two types. You have smooth ER, which we'll talk about the example why in a second. So what smooth ER does is it does not have any ribosomes. Okay, hence the smooth. So no ribosomes. So it's not making proteins there. What it's meant for is it produces or synthesizes of fats or lipids. And so the reason why I showed the sundae is because sundaes will make people fat if they eat them all the time. So when you make a sundae, you're making fat. So same thing, just a stupid way to remember it. What also it does is it detoxes drugs. So it breaks down drugs and poisons. So cells in the liver would have a lot of smooth ER because that's what it's going to do for its function, is it detoxes your body. Rough ER, so this has ribosomes. That's what makes it rough. Boom. So what it does is, really for these ones, all you need to know is that it's just a channel where proteins travel through. Okay? Before something else happens. There's lots of other processes that occur, but they're going to be a little bit more advanced. We'll see those in Bio 12 and in university. Oh, sorry. And the reason why I showed this is because these are the different tunnels, different channels, just like ER. Okay, so we're almost done here. We have the Golgi apparatus, also known as the Golgi body or the Golgi complex. This is where ER products, so lipids and proteins, are modified, stored, and then shipped. So modified usually means that something's added to them to help them better travel or better able to locate where they're going. And the way that it does that is through transport vesicles. So these little vesicles bud off of here, and they actually budded off of the ER too, and they travel to their certain areas in these vesicles. Cisternae is just a word that you need to know that just means the flattened membranous sacs. So these here are your cisternae. And then finishing off, these vesicles that transport sometimes can transport enzymes. And if it's an organelle that transports digestive enzyme, it's called a lysosome. It pretty much goes around, well, we'll see. We'll see here in a second. So the reason why I showed this is that this is plastic surgery. These are pretty much manipulating all your proteins, okay, or changing your fat, right? They're modifying your lipids and your fats, or lipids and your proteins, just like plastic surgery does. Okay, last one, I think, lysosomes. Whoa, that was quick. So what lysosome does, it's pretty much the vacuum cleaner of the body. So it has hydrolytic enzymes, which means that it breaks down products. Hydrolytic enzymes. So it digests big molecules. And we call those macromolecules. Yikes, nice spelling. What it does is it pretty much ingests it. And it's through a process called phagocytosis, which we'll look at later. And so the example here is the vacuum cleaner. So you can imagine what happens if you never clean your house. If you're never cleaning out your cells, things build up and break down just like it would there. Okay, so for us, making sure we just know all of the functions. But really, I would try to remember or create your own some analogies. So ways to remember it, because if I say Golgi, you got to find a way to remember that it's something that modifies proteins and lipids. Okay? Have yourselves a wonderful day.